Hello, my name is Konstantin, and today I'm going to tell you about new neural network AlphaGenome, which we're thinking about to use in Oxford University for NGS data interpretation. I'm working at the Department of Oncology, so I will tell everything in connection with this field of knowledge and medical practice. AlphaGenome is a new deep learning model that was introduced just in this year, and it's working on a huge fragments of DNA. It's able to process about 1 million bases, and the most important thing that it can predict several types of regulatory signals, not only single variants or not only specific types of structural changes, but it's able to tell us about how certain gene expression level will change, how splicing will be affected, transcription factors binding, histone modifications, chromatin contacts, and so on. Why do we need this? In our context, we're working on projects which are in most of the cases using cell-free DNA for cancer prediction, and it's extremely important to understand whether the mutation found really changed something or it's just a random noise which we shouldn't bother to analyze. So AlphaGenome helps to link that sequences to a function of a certain gene. So today I'm going to talk about general concepts of cell-free DNA analysis, how we're using them as markers for mutations, and how we're able to utilize methylation and fragmentomics to provide certain level of certainty in our predictions in cancer diagnostics. I will highlight some of the projects which could be found on our webpage, and I will provide a short glance on AlphaGenome architecture, how it's working, which layers are there, and how it could be compared with previously used tools like Informer, Splice AI, or Borzoi. And I will show how predictions made by this model could be utilized in practice, what are they, how they will look like, and how we can build them inside in our cell-free DNA analysis pipelines in cancer prediction. So just a short summary, what is a cell-free DNA and how it's being used. It's just a pieces of DNA that circulate in our bloodstream. In cancer patients, some of those fragments will originate from tumor cells, and taking those fragments is being called liquid biopsy, and there are several approaches how to utilize them for early cancer predictions and diagnostics. Basic things like checking for mutations and single variants, substitutions, insertions, deletions in DNA. We can analyze methylation when methyl groups on cytosines, adjust the regulation of certain genes if they are being expressed or not in certain tissues, in tumor tissues. And also we're using so-called fragmentomics when our predictions are based on cell-free DNA fragment size and how exactly those fragments were formed could also lead to ideas how chromatin is packed in cells, and it's also related to oncogenic processes. And the problem is that on early stages of disease, we have only very little amount of tumor DNA. And our biggest problem is to divide those signals from that tumor DNA from normal DNA, which is normally circulating in our bloodstream. So it's quite interesting idea to combine multiple signals into one predicting pipeline, which will analyze multiple factors to provide a better result. One of the key sources of information in cell-free DNA is methylation. In tumor cells, it will be disrupted. For example, promoters, which are regulating certain regions, can be hypermethylated, and it will lead to silent genes, so certain proteins expression will be prevented, and it could lead to cancerous process. What's important is that methylation profile in cell-free DNA reflects the fabric of origin of the DNA. So we are able to tell where cancer is located. And it's a good addition to the fact itself that there's a chance this patient have a cancer, because we will need clinical confirmation for this diagnosis. There are different ways to determine methylation, and the one used at Oxford is TAPS, Data-Assisted Pyridine Variant Sequencing Technology, a method that allows us to gently read methylation without destroying DNA, and in this way we get both sequence and methylation marks at the same time. Another approach is fragmentomics, where we determine how exactly DNA is being cut into pieces in our blood, and healthy and tumor cells will present different patterns of this fragmentation. For example, length of fragments normally will be around 167 base pairs, and tumor will provide more short fragments in general. The motifs at the ends of the fragments are also important. They depend on whether the nucleosomes, the packaging of DNA histones, were located, and how enzymes that cut the DNA worked with them. And it turned out that those signals can distinguish a patient with tumor from a healthy person with high level of accuracy. So we're able to find about 0.9 area under curve in such a studies. In addition to methylation and fragmentomics, there are other sources of information. For example, Polygenic risk score is an individual risk score based on tens of hundreds of single nucleotide polymorphisms, and it's not a basic genetic prediction based on one nucleotide, but it's more like a more general approach covering multiple structures. So structural variants and breakpoints are major regions of the genome. So there could be translocations, deletions, integrations of viral DNA, and it could be also noted from cell-free DNA. And as a result, we have a huge amount of epigenetic marks, such as histone modifications, and we are not able to measure them directly from cell-free DNA, but we can integrate multiple evidence and use them as part of the process, combining methylation and fragmentation patterns all together with other types of biomarkers. So as a result, we have multidimensional structure which is able to provide us better predictions and better insights. At Oxford University, there are several initiatives which are working in this field. For example, TriOx is a test which is aiming for multiple cancers, and it's utilizing methylation for prediction of a chance of having a tumor DNA in that patient. Scan pathway is a project for patients with non-specific symptoms where DNA was collected, biobanked, and it could be used as early markers if at a certain point of time we will be able to confirm that that patient developed a cancer. So we're gathering data to be able to understand that in future. Explored working with cohorts of precancerous blood disease, and it's studying mutations and epigenetic shifts, which is able to predict progression of the disease. Aerial is a project in Africa where we're implementing cell-free and circulating tumor DNA tests for lymphomas caused by Epstein-Barr virus. All those projects need interpretation tools, and AlphaGenome could be an interesting option as an addition to our current pipeline, which is already being utilized in those projects. So let's talk about AlphaGenome and what is it and how it's working. AlphaGenome takes a huge chunk of DNA as input. As I told before, it's 1 megabase or 1 million base part of DNA. It's more than 8 times bigger than previously used models, and based on that data, we're able to receive much bigger variety of signals, which we can use later to distinguish a healthy sample from tumor-related samples. So it will be RNA sequencing, which resulting in gene expression, pH transcriptions, splicing, chromatin openness, histone modifications, binding sites, chromatin 3D structure, and so on. So that this new model can predict almost everything that can be utilized in determining how certain genes are being regulated in our DNA. From the inside, AlphaGenome is something like a hybrid of UNet and transformer models. First, convolutional neural networks are reducing in dimensionality, the same as it was done in UNet. Then central unit is a large transformer with attention mechanisms, which processes connections over long distances in that sample. So it's important because we're providing a long sequence as an input. To pull the DNA megabase, it's dividing it into 8 pieces of 128,000 bases. Each piece is processed on a separate TPU, and there is communication between those parts, so common features will be persisted in that analysis. Next stage will be decoder, again increasing the resolution of the nucleotide. And there is also a special paired-wise block that simulates interactions between positions, which is necessary for predicting chromatin contracts. Let's take a look at a bit more details how it's working. So encoder is taking a long DNA sequence and compresses it. It uses layers that are good in finding patterns, convolutional blocks. They are convolutional blocks, and they are reducing the size of that data, so it's pulling. As data gets smaller, the model adds more detail to what it's looking for, so it's increasing feature channels. It shrinks the sequence resolution from 1 base pair down to 128 base pairs. Then we have a transformer tower. At the narrowest point of the funnel, the model uses powerful components which are able to compress data to understand relationships within those sequences. And at the same point, model starts creating a map of how different parts of the sequences are related to each other. So it's keeping a higher structure as well. Next we have decoder. So after processing, the model needs to return to its original size, and decoder does this by revising the process of the encoder. So it's a similar process which is working as in most transformers both ways. It uses layers to expand the data like in upsampling, so it's getting back to 1 base pair resolution. And it gets a little help by pulling information directly from encoder at each step, and it's using skip connections to ensure it doesn't lose important details during the expansion in this process. Next we have the final layer, the output embedder, and this is the final step where model tides up the data before sending it to a specific task, like predicting a gene or mutation or its functionality and so on. So basically the model takes a huge DNA sequence, shrinks it down to its most important features. It's quite similar to convolutional neural networks and transformers with attention mechanisms. It's analyzes those features and then rebuilds the feature sequence to give a detailed result for a specific task. Think of it like summarizing a book, so it's quite similar to large language models, but it's working with different type of data as an input. AlphaGenome has been trained on data from large government research projects and consortia that had experimentally measured various DNA proportions and properties related to certain features of humans and mouse cells and in relation to their tissues. Most important sources are ENCODE, an encyclopedia of DNA elements. The goal The goal of this project is to identify all functional elements in human and mouse genome. Another data input was from GTEx Genotype Tissue Expression Project. This project provides publicly available resources for studying gene expression and regulation in a variety of human tissues. Another one, 4D nucleosome. This consortium studies the three-dimensional organization of cell nucleus and its role in gene function. Another one, Phantom 5, mammalian genome functioning annotations, and this project uses gene expression analysis to map transcription onsets, pointing and measuring the expression of a huge number of human and mouse genes. These datasets contain thousands of genomic tracks or molecular properties that are model used to study relationships of DNA sequences of gene regulation and other biological factors. The model was trained to simultaneously predict those properties in a variety of modalities, including gene expression, RNA splicing, and so on and so on. So, just as I was telling before. So, training was quite complex. It was divided into two stages. First, they make teacher models. They take different pieces of genome folds and train their own model on each of them. In addition, there was one model covering entire genome. And next stage was distillation technique, when we have a student model which is trained based on teacher model data, and it's being used to make an average behavior from those teacher models. So it will be combining knowledge from multiple models trained on different data. And in addition, it allows to compress that knowledge into a single network, which is enabling it to be much faster than a combination of that separate teacher models. Another important point will be that they were trained not only on human data. Using data from mice was important for the model to better learn general patterns in DNA regulations and not to stuck with human DNA only and human regulations only. In terms of data modalities which were used for training, they were quite simple. In DNA sequences, it was just nucleotide letters converted into one hot encoder numerical arrays, and for molecular properties it was a bit more complicated, and it was as a numerical arrays representing the signal of value from an experimental assay and its corresponding DNA base pairs. So it was like data matrices with numbers. AlphaGenome predicts 11 types of data at once. It will be expression, splicing, domain openness, histone modifications, expression factors binding, 3D conformations, and so on. In terms of accuracy, the model is ahead of all previous solutions in 22 out of 24 benchmarks. For example, it's better than Informer on expression and openness, and better than SpliceAI and Borzoi on splicing. At the same time, the prediction of one option takes about a second, on a good GPU, and this can be really used in practical terms, so it's not like a tool for scientific study, but it's ready to be incorporated into clinical pipelines. There were several models which were covering that area before AlphaGenome. They could be divided into two major camps. Short context ones like splice AI that could work with on one single nucleotide level, but they were able to process only not more than 10,000 base pairs around that nucleotide. And another type was long context models like Informer or Borzoi that were taking tens of hundreds of thousands of bases, but they were using average signals, not going deeper to the level of single nucleotides. So there was always a challenge: are we going to sacrifice some data in generalization, or we will be able to cover just a part of DNA fragment which could be affecting certain gene in its function, expression, and so on. It's not that easy to understand how exactly it's being evaluated. So what does it mean to evaluate a certain predictive option in that algorithm? We're taking a sequence with a reference allele and alternative allele, so it's our variant. And we're running that variant through the model, and we're comparing predictions from our new model and previously used model. So it will be comparison from AlphaGenome with another model. For expression, we're able to calculate fold change as a certain number which was provided by that models. For splicing, we're looking at how much the probability of using a site or connection exons will change. For openness of histones, it's just the same as for splicing. So we have several metrics which could be summarized into delta score, which could be used for that evaluation of different models. AlphaGenome can predict both the use of donor acceptor sites and the number of specific junctions in genome. This is important because many diseases are associated with splicing disorders. In the article, it was provided as example with U2SURP gene, and the variant in the donor site leads to the fact that splicing is simply cut off, so it's not being performed, and the model is able to correctly predict this in that sample. The same principle is applied here for splicing variant scoring, and we are comparing reference and alternative variants, and we are taking maximum chance in the probability of using the site for splicing. In the same U2SURP example, an important motif that CAG trinucleotide can be visualized as being used. If we break it, the model shows a sharp drop in signal. So if it's just related to certain trinucleotide existence. So we're able to say that change in a certain nucleotide is dangerous in terms of it will affect splicing, and as a result could have a disease progression. So AlphaGenome significantly outperforms SpliceAI, Pangolin, and Borzoi in tests for pathogenic variants. It copes especially well with complex cases, for example, mutations in deep introns where the influence is far from usually tested sites. So it's just a wider effect. And in short, this model is quite effective in predicting effect from mutations in introns and how they're affecting splicing in general. When we're talking about how certain variant will affect expression, we are looking at how much RNA will be produced after certain changes in variants. After that, we are able to calculate how the changes will be able to affect transcription. So if it will affect protein structure as a result of changes in RNA. As a result, we can estimate for a specific gene how exactly its expression will be affected, even if there are no changes in variants related to exomes of this gene. So it will be regulatory fragments. In the paper, they provide a good example for a variant in EPOL4 gene on chromosome 22, which affects expression. AlphaGenome predicted that allele replacement did boost gene expression, and this coincides with the results of GTEx expression quantitative test and AlphaGenome correlation of predictions are the same for that experiment, and it's higher than predictions from Borzoi. For example, it was 0.5 with AlphaGenome and 0.39 with Borzoi. In addition, it could be used for in silico mutations, so we can make some virtual changes and substitutions in genome and analyze how that motifs are affecting promoters and response in the effects of that gene. If we're looking at large-scale comparison, AlphaGenome has a prediction correlation with EQTL test with 0.49 and Borzoi has only 0.39, so it's a bit better. And if we're taking a confidence test, AlphaGenome can correctly predict the direction of the effect almost twice as often in comparison with other tools. This means that we can use the model to help to understand which of the variants of genome-wide association study are related to causes of a certain disease in such a studies. So we have a lot of data related to DNA sequence and clinical data from that patients, but it was impossible to prove, it was hard to prove that certain motifs in genome are causing certain disease. So analyzing regulatory mechanisms will allow us to do that. Another task is to understand which enhancer control which gene. So AlphaGenome allows to do a zero shot. We will look at whether a mutation is an enhancer and if it affects gene expression. In tests, the model outperforms Borzoi for such tasks, especially for long-distance regulations. So if a certain regulatory fragment is further on a DNA than 10,000 base pairs which were used before, it's impossible for them to be detected with previously used tools. And we can use these predictions as a feature for standard algorithms which are being used before AlphaGenome, so it could be incorporated in addition to our existing tools. The model is able to predict which poly A site will be selected, proximal or distal. This affects the length of the non-translatable regions in DNA. And there is a separate cover ratio matrix which also provided as example in the article. In PMP22 gene, AlphaGenome is better at predicting the ratio of proximal and distal transcripts than Borzoi. In terms of polyadenylation quality tests, variance affecting correlation is 0.89 versus 0.79 in Borzoi. In addition to expression and splicing, AlphaGenome predicts chromatin openness with ETAC sequence and DNA base tests. It links transcription factors, histone tags, and 3D chromatin contractions, and this data expands the picture. For example, it's possible to understand whether a variant affects the availability of a promoter or the contact of the hunter with another gene. A very useful tool. It could be used to virtually predict changes in certain nucleotides and how they will affect expression of a certain gene. So it could be used for in silico mutation simulations, and it could be easily analyzed a certain variant if we even don't have that sample on practice. On this slide, I'm showing pseudocode of how AlphaGenome is being used. So we're just taking segments around a variant, and we're using both data for reference and alternative allele, and providing that data to the model for the prediction. Then we can calculate data for expression and other changes like splices and differences in likelihood of using a certain site, and it's quite easy to be embedded into the pipelines which we're currently using. And the most interesting thing is that we can combine different metrics together. For example, in wide registration studies, we're analyzing user features from different sources, and if we're adding AlphaGenome predictions by expression, splicing, openness, and so on to such a classifier, we will be able to increase accuracy. So the article shows that the simple random forest on AlphaGenome features is ahead of all previously used approach to predict causal expression quantities in certain genes. So the model is able to provide us a set of features which we could use in our analysis pipeline later. AlphaGenome could be used to prioritize variants from wide genome association studies, and the model shows which SNPs actually change regulation and which are not changing it. In the tests, it was possible to close almost half of all credible sets that are using for functional explanation. In the context of cell-free DNA, this means that even rare variants from liquid biopsies can be evaluated in terms of their regulatory effect. If we embed AlphaGenome into cell-free DNA analysis, the next steps will be like, first we will have to process data normally for variant calling, and we'll find single nucleotide variants and deletions, so called small mutations, and for each option we will be able to calculate and produce scorings in AlphaGenome. We can filter only that variants that have a stronger effect, for example, variants which are disturbing splicing or structures of our DNA conformations, and for top candidates we are able to process validation with different methods. In such a way, we're able not only to record mutations, but also immediately understand which of them are functionally significant. Although AlphaGenome doesn't directly predict fragment patterns, its outputs can be used in interpretation of fragments. For example, if the model shows that the region is open, then we can expect that more short cell-free DNA fragments will be produced by that region. Similarly, if transcription factor binding is predicted for that region, this may explain the motifs at the ends of the fragments. In this way, we can link AlphaGenome's predictions to real-world fragmented data from cell-free DNA. Also, AlphaGenome itself doesn't predict methylation, but its predictions for chromatin and binding sites can be integrated with methylation data. For example, in TAPS, we're showing promoter hypermethylation, and AlphaGenome shows loss of the insulator site. This straightens the regulatory failure hypothesis for certain region. Such data synthesis allows us for much deeper understanding of what exactly changes in tumor cells due to certain mutations and fluctuations in methylation patterns. For example, in TriOx, where we obtain both sequence and methylation at the same time, we can see a region with an altered methylation level. With the help of the AlphaGenome, we can assess which genes will be potentially regulated and predict the effect on expression from that genes. Or the other way, we are able to find rare variant and immediately see how much it changes the regulation for that region. In this way, AlphaGenome could be used as an analytical model that complements our multimodal approach, which was already implemented. In scan pathway project, we are collecting cell-free DNA from patients with non-specific symptoms, and often such people are referred just in case, and if it's very important, it could be very important to quickly understand whether there is cancer or not. If we're finding rare variant in cell-free DNA, AlphaGenome can tell us if it's just a random mutation or it's actually disrupting the function of a certain genes. For example, if a patient is found to have a mutation in a donor splicing site in a tumor suppressor gene, the model will show a strong effect. Such a signal will help the doctor to understand that the case is serious and requires additional examination. For a real project where we study Epstein-Barr virus-associated lymphomas, in such patients, cell-free DNA contains not only human fragments but also DNA from Epstein-Barr virus. AlphaGenome could be adopted here, for example, to create a reference where the viral and human genomes are combined together, and could be used to evaluate the impact of Epstein-Barr virus integration sites. Even though it's possible to analyze the human areas where the integration and see the viral fragments breaks and how important those breaks are on regulatory elements, this will give us an understanding on the mechanisms of virus-induced cancerogenesis. Oxford is a study of precancerous blood diseases where patients often have clonal mutations, but they not always lead to leukemia. If a mutation disrupts an important regulatory site, for example, drastically reduces the expression of a suppressor gene, this could be a predictor of a progression. AlphaGenome allows us to evaluate this by giving a functional annotation to the found variants. Thus, we can distinguish patients with a high risk of progression to a malignant form. The article gives us a very illustrative clinical example. A patient with T-cell leukemia had a deletion near tau1 gene. It removes the binding sites, CTCF, and AlphaGenome predicted that such a deletion would lead to an increase in tau1 expression. This coincides with known mechanisms like the loss of the insulator enhancer begins a pump tau1 and it's activating the oncogene process. And this is demonstrating of how the model can explain driver rearrangements in the tumor genome. In polygenic risk score, it could be used to measure the risk based on many single nucleotide variants. So AlphaGenome could be used not as a directly tool to calculate that polygenic risk score, but it's possible to integrate with existing tools and to analyze multiple variants in such a patients and combine that data with the model. So AlphaGenome will give functional changes, and we're able to calculate polygenic risk score not based on initial nucleotides, but on AlphaGenome predictions for those nucleotides. Also, we can combine both approaches and provide better prediction, combining not only data from DNA variations, but also from effect variations. So it will be DNA effect variation risk score. In cell-free DNA, we can sometimes catch DNA junction breakpoints, and this is the result of rearrangements, translocations, or even integration of the virus DNA, and AlphaGenome could be used here for annotation of the data. We can understand next to the breakpoint nucleotides, and the model will tell us which regulatory elements are affected by the changes related to a certain breakpoint. It's possible to predict which gene will be activated or otherwise turned off. This way we can turn raw data into biologically significant hypotheses. Of course, there are limitations for the model. It's trained mainly on normal cell data. Tumors could have completely different epigenetic profiles, and this means that we need to validate that model on tumor data. Also, it could be additionally trained on different types of disease, so it will be significantly improved in quality of prediction for such a disease. Methylation isn't directly predicted by the model, and it's a separate task. And the training was conducted on a limited set of tissues, so predictions may not be always the relevant ones for specific types of tissue, specific types of tumors, and so on. The model itself is quite big, and it requires serious computing resources to perform. For now, it's able to be used as API, and it's available for scientific studies. In the future, it's logical to train the model on the data from methylation, from tumors, and so on. Clinical tests are often limited to small panels of genes, and if we will add AlphaGenome layer to those panels, we can interpret even the non-coding variants which could be previously ignored. So it will drastically increase results from our panels. So as a result, it will be able to make our predictions more useful for doctors and our patients. If we want to work with AlphaGenome for now, DeepMind provides examples and training materials on GitHub, and it's quite generous, and from that data it's possible to implement that model into our pipeline. So all the data on which the model was trained were open tracks from ENCODE and other epigenomics projects, so it could be freely used for research projects. So practically there are no barriers to integrate this tool into existing pipelines and existing processes. To summarize, AlphaGenome is a first model that can simultaneously see megabase context and make accurate predictions at the nucleotide level. It outperforms all previously used solutions in terms of accuracy on expression, splicing, polyadenosine predictions, and other tasks. For cell-free DNA analysis, this means that it could not only find variants, but also able to understand their functional effect. In combination with our existing projects, it could be used as a tool to improve their results, quality, and how they are being performed. So AlphaGenome is not just another model, it's a platform for functional interpretation of genome data. And in oncology, it's quite important because it's a really complex matter to understand how changes in regulation of genome are related to a certain disease. Thank you for your attention.